In our last module, we talked about the scientific method, and we discussed the various steps in the scientific method and what a hypothesis is and why it is so important in the scientific method. Just as a review, all experiments start with an observation or a description of a phenomenon and a need to investigate why that phenomenon is occurring or how it is occurring. We will come up with a hypothesis to explain what we think is going on, and we will predict and test that hypothesis. That hypothesis must be a statement, and it must be very specific and testable. Following our predictions and our experimentation, we will analyze our results and come up with a conclusion, and subsequently there will be independent verification of our results, and other people might come up with their own conclusions as well. So in this section, let's talk about experimentation. The experiments need to be designed very carefully so that their results can be interpreted properly. You can design an experiment poorly and modify too many variables at the same time, and at the end of that experiment, you're not going to be able to explain what is going on because you've changed too many things at once. So therefore, it's very important to change one variable at a time in an experiment. So in a very simple experiment, we might have one experimental variable and one control variable, so only one element that we are changing at a time. And we're going to use that to study how that variable affects the phenomenon that we are looking at. For every experiment, we're going to make some assumptions. We need to assume some things are going to happen according to, the laws of physics or, other biological rules that we are just going to assume are going to be behaving properly. And we're going to come up with specific procedures. We're going to, in advance, create an outline of exactly what we're going to do, and then we will follow them. Okay, so let's look at an example. I think the best way to learn is to look at examples, and that, I hope, will give you a very clear idea of the scientific process. In this case, we're going to be looking at poisonous snakes and how evolution has affected their presence in certain areas. We're also going to practice designing a hypothesis once again and analyze the results that were presented from this study. Okay, so what's our observation? What's the first step in the scientific method, in this case study of scientific inquiry? Someone observed that there are many poisonous snakes that are very brightly colored, and we assume that this means that their bright colors are warning potential predators. So these snakes have evolved to have very bright red and yellow stripes, and predators know not to attack these snakes because they are poisonous and something catastrophic might happen if they attack these colorful snakes. And this has been learned through evolution. So predators have learned not to attack these bright snakes. Because these brightly colored snakes, survive better because of their coloring, other snakes that are not poisonous have evolved to have bright colors as well. So these are known as mimics. They are not poisonous, they are harmless, but they look a lot like the poisonous species. Henry Bates hypothesized that this mimicry in the colors evolved in harmless species as an evolutionary adaptation that reduces their chances of being eaten. So that's a strong hypothesis that's testable. It's a statement, and we can look at what happens. So let's design the experiment. This hypothesis was tested with the poisonous eastern coral snake and its mimic, the non-poisonous scarlet king snake. Both species live in the Carolinas, but the king snake is also found in regions without poisonous coral snakes. If predators inherit an avoidance, or we don't know if the avoidance is inherited, or that's something that would probably require further investigation. But if the predators learn to avoid the coral snake's coloration, then the colorful king snake will be attacked less often in regions where coral snakes are present. If the brightly colored snake, the non-poisonous one, is present in an area, so all of this orange area without the lines, are areas where the non-poisonous colorful snakes are present on their own. So if there are no poisonous snakes present, and these are present, the poisonous snakes are present in the dark colored hashed area here, so both species are present here, then the predators in this area might not learn to avoid those snakes due to their bright coloring. So they have not encountered the poisonous snakes, and they're not going to learn that that colorful pattern means avoid that type of species. You might start thinking about how to design an experiment now. We need to test whether or not these non-poisonous, brightly colored snakes are going to be attacked more frequently in the area that doesn't contain poisonous snakes. All right, so now you guys are budding ecologists. Let's design this experiment. So we have an idea of what to do already, right? We want to test how frequently these kind of snakes are attacked. And let's think about how we might do that. How might you design this experiment? Why don't you pause and think about it for a couple minutes? It would be very difficult to do this experiment with real snakes. We'd have to tag a lot of these snakes and follow them over time, and that might be kind of difficult with these kinds of animals. We do that with often larger animals. But let's design an experiment that doesn't require us to use the living species. All right, so let's take a plastic snake and something that is made out of a soft material so that we could see how many times it was attacked. So we can see bite marks or any other signs of predation that fake snake might encounter. Okay, so let's do it. Let's take plastic snakes that look like this, and we'll put them in both experimental locations. We'll put some in this north area where there are no poisonous snakes, and we'll put some in the south area where poisonous snakes are present. And then we'll measure which group was attacked more. That sounds great, right? So we have two different conditions. We have these snakes in two different locations, and we have a quantitative measure that we are actually going to be able to look at. So we're going to measure how many times these plastic snakes were attacked. That's a hard number, right? That's actual quantitative data that we can start comparing. All right, let's think a little bit more about what we might want to do. Let's say that these colorful snakes are just bitten more frequently because they're very noticeable and obvious, and maybe there's some other reason that they might be attacked more frequently. Let's add another experimental condition. Let's take some fake plastic snakes that are not brightly colored and put them in both regions and that will be our control population, and we'll see how many times the non-colorful snakes are attacked. Okay, so this is what they did. To test this mimicry, researchers made hundreds of artificial snakes. They took an experimental group that resembled the king snake, so they had that bright orange pattern. They took a control group that resembled plain brown snakes, so these were not brightly colored, and we can compare how many times the brightly colored group was attacked and how many times the plain brown snakes were attacked. Equal numbers of both types were placed at field sites, including areas without poisonous coral snakes. So this is the experimental design coming into play. The researchers thought, here's the conditions that we're going to use. Here's our experimental design, right? We're going to take all of these plastic snakes. We're going to take brightly colored ones, non-brightly colored ones. We're going to put equal numbers in regions that probably are similar, right? Regions that have similar conditions. And then they decided how long they're going to let the experiment progress and how they are actually going to measure the data. So when they went back and got the snakes, so let's look at the results or the pictures of the results. When they went back to get the snakes, they saw puncture marks, right, wounds of from other animals that had attacked these snakes. And now, hopefully in the beginning of the experiment, the researchers decided in advance how to quantify this data. So how would you quantify this data? What would you do with these injured plastic snakes? One thing you might want to measure is how many times they were bitten, or you might measure the size of the artificial wound, or the size of the wound in the artificial snake. You might want to measure, right, the depth or the radius, the number. So these are all quantifications that the researchers need to be able to compile their data and compare the two groups. So here's the artificial king snake, here's the artificial brown snake, and we see that the brown snake has been attacked multiple times and it looks to be in pretty bad condition. So they decided to follow the conditions, the experimental and control conditions, for 4 weeks. After 4 weeks, the scientists went and retrieved all the artificial snakes, and they counted bite or claw marks. Then they analyzed their data, and they graphed it, and their data, the results, fit the prediction of the mimicry hypothesis. The ringed snakes were attacked less frequently in the geographic region where coral snakes were found. So the brightly colored snakes were attacked less if the poisonous snakes were physically present. And here's their data. Because they designed the experiment in a way that allowed them to get quantitative data, they were able to create measurements and bar graphs to demonstrate their data. Here's the north region in the Carolinas, where the coral snakes are absent, and we could see that the artificial king snakes were attacked much more frequently than the brown snakes. However, in the south regions where the poisonous coral snakes were present, the artificial king snakes, which were brightly colored, were attacked much less frequently, and the brown snakes were attacked much more frequently. And of course, there's other measurements that we discussed that the researchers could display, right? The number of attacks of each snake and the severity, etc., etc. So in this experiment, they had a very important, they designed it to have a very important control, right? So here the brown snakes are the control. And without the brown snakes, we'd be able to say the artificial king snakes were attacked much less frequently in the south, where the poisonous snakes are present, but we wouldn't be able to make the same conclusion necessarily. So what if in the south there are less predators in general? What if there's some other reason why there might be fewer attacks that has nothing to do with the color or the poisonous abundance of poisonous snakes? So our brown snakes let us demonstrate that in fact there are predators in that area. The brown snakes are being attacked. However, the difference in the number of attacks is in fact, or seems to be, due to the coloring of these artificial snakes and whether or not the poisonous snakes are present. So in this experiment, there was an important control. A controlled experiment compares an experimental group, so in this case, the artificial king snakes, the colorful snakes, with the control group. Ideally, only the variable of interest, for example the color, would be different between the two groups. So we would not want to start changing around a lot of different parameters, because that might convolute our final results and make it impossible to come to a conclusion. The controlled experiment means that control groups are used to cancel the effects of unwanted variables, right? So the abundance of predators might be an unwanted variable, right? The climate, we're looking at two different locations, so that a lot of things might be different between the two locations. A controlled experiment does not mean that all unwanted variables are kept constant. We're looking at an outdoor area. Obviously, things are going to be different, but we want to try to minimize that difference as much as we possibly can. In the lab, we also might have differences but we try to keep them to an absolute minimum. So, for example, here's an experiment that was done very early on by one of the first scientists that we know of in human history, where they were trying to disprove whether or not life spontaneously arose. So people saw that if they left meat out, for example, that meat after a long time or after some time might start growing maggots, and those maggots would hatch into flies at some point. So during this time period in human history, it was hundreds of years ago, they decided that life spontaneously arose, that from this meat, flies were spontaneously arising. One guy was smart enough to say, you know what, I think that life is not spontaneously arising, so I'm going to cover this meat with a cheesecloth and show you that the flies actually are not spontaneously arising from the meat, and they are actually coming from an external source. So he set up 2 jars. He set up one jar that was open and one jar that was covered with a cloth, and demonstrated that the flies did not spontaneously arise in the covered experimental group. So for every experiment, you need a control. Controls need to be carefully designed and are the most important part of the study. Okay, let's finish by talking about theories for a couple minutes. So, like we said in the first section, there is no absolute truth in science. Science is 99.99999 sure of a result or of a conclusion at any given time, but never 100%. And we take all of these results that, we are almost completely sure of, but not completely sure of, and we combine them together, and we take information from physics and information from, all of the history of knowledge that we have and different hypotheses to build a scientific theory. So theories are large, overarching ideas that globally scientists can agree on. In the context of science, a theory is broader in scope than a hypothesis. It's general and can lead to new testable hypotheses, and it's supported by a large body of evidence in comparison to hypotheses. So theories are developed from observed facts, right? So the theory of relativity, right? For example, we know there's a lot of observed facts and then a lot of math and physics calculations that went into it, and a lot of experiments that came together to basically demonstrate that the theory of relativity is a strong theory that we are going to utilize as if it is a fact. Hypotheses are informed guesses developed from existing facts. We can use data and observe facts and hypotheses to create models. So a model is just a theory, right? A flow chart of how we think something works. Laws, descriptions, often mathematical, of natural phenomenon. And together, hypotheses, models, postulates, and laws come together to develop theories. Scientific theories are not hunches or opinions. We can consider them basically to be truth. But as good scientists, we know that there is no absolute truth. But we will take them to be as close to truth as you can possibly get. Scientific theories are the ultimate goal of science. They are not dogma, right? They are not based on a collective opinion. They are based on scientific facts. But they can change as more data accumulates. So for example, Pluto, right? At one point we decided collectively that it was a planet, and then scientists accumulated additional data and decided that maybe it did not fit the criteria of being a planet anymore. Theories represent scientific consensus. Science uses a lot of deductive reasoning. So, for example, if organisms are made of cells, and humans are organisms, then humans are composed of cells. So if we see one thing, and that's linked to another thing, or if 1 is 2 and 2 is 3, then 1 is to 3 in the deductive reasoning method. You can read more about that if you like. However, because we might be making correlations between two things that are not necessarily correlated, it's important to remember that correlation does not always equal causation. Events, any event, right, can be coincident in time or in space. A correlation between things is easy to establish and very easy to abuse, and we'll see some examples of that. And this would be considered a logical fallacy. If A precedes B, then A caused B. But it's very important as scientists to remember that correlation does not equal causation. And this is a pitfall that scientists can fall into frequently when they are interpreting their results. Okay, so for example, here's something that looks quite correlated. The fresh lemons that are imported from the USA to the USA from Mexico, and the number of highway fatality rates. So as we increase lemon import, the number of highway deaths goes down. So you might be able to think of some reasons why these numbers correlate, but of course, one has probably absolutely nothing to do with the other. Here's another example. The abundance of Internet Explorer, the web browser use, right? The proportion in the population that of people that use it, when compared to other internet browsers, and the murder rate in America. And we can see that there's a very strong correlation, that as Internet Explorer use went down, so did the murder rate. So does that mean that using Internet Explorer might make you fly into a murderous rage? How about organic food sales and autism? Here's a good one, right? So we can see that they're very correlated. This number here, the R value, is a quantitative measure of correlation. But we might think that the number of organic food that's being sold in America and the rates of autism might not necessarily be connected, right? We don't think that organic food causes autism, most likely. In all scientific studies, there's going to be probably some statistics to demonstrate that there is that the probability of two phenomena being linked is well below the chance of them being linked through random chance. So talking about statistical significance is just a way to mathematically quantify the likelihood that a relationship between two or more variables is caused by something other than random chance. And of course, this can be abused as well, right? The previous examples were all statistically correlated and had very high correlation values. But this does not necessarily mean that they are causal. So statistical significance does not always mean that our conclusion is correct. Statistics can be misused and manipulated. You are always looking at a small part of the population, almost always, right? I guess in some cases it is possible to measure every individual on Earth, but that is almost rarely the case. So usually we're only looking at a fraction of the population. This can lead to measurement bias, right? So when you look at the effect of a drug on a specific population, it's very important to try to control for a lot of different elements that might affect the results. So gender, race, background, economic standing, right? These are all things that can affect people's health and affect the result of how a drug might affect their health in some way. For example, election polls, right? Exit polls often, or not always, but sometimes don't mimic the results of the actual election. So it's important to remember why did the results, why they might be skewed in a certain direction, and to remember that might not represent the entire population. So accuracy and precision, right? Accuracy in scientific experimentation, accuracy of your data is how close the observed values are to an expected or standard value, and precision might be the deviation between experimental values. So you might have very high precision, right? All of your values might be very, very close together, but they might not be accurate depending on the way that you designed your measurements. So if you are measuring a very small part of a population, let's say you're looking at the effect of a drug on a specific disease, and you take all of your subjects for that experiment from one family, you might have a lot of precision, right? All of your results might be grouped very close together, but the accuracy might be quite low because you've selectively taken a small part of the population to do your experiment with. Okay, so that is what we have to say about experimental methods. I hope that you've taken away some information about how to design an experiment and the steps that an experimentalist takes. And of course, we remember that it's very important to design your experiment in advance. That makes it possible for you to design it in a way that you will be able to interpret the results as accurately as you possibly can. And of course, it's important to remember that every experiment is looking at a subpopulation of a larger population, first of all. Second of all, that correlation does not equal causation. So because we make a lot of assumptions in experiments of what else we think is going on, we need to always keep an open mind about our interpretation. And as scientists, we present the results as they are, as accurately as we can. And we can present our conclusions, but it's possible that other scientists or individuals might come up with different conclusions than the ones we made. And that's okay. That's all part of the process.